my colleague, Dr. Rim Kefi. She's the first anthropologist, molecular anthropologist in North Africa, and she is a team leader in the biomedical genomics and oncogenetics laboratory in Institut Pasteur de Tunis. And the floor is yours, Rim. She will speak about DNA and ancient population. I'm sure you will enjoy her talk. Thank you.
Thank you very much, Alfa. Good afternoon. Hello, everybody. Thank you for your presence here. It's a great pleasure for me to present this talk on ancient DNA study of North African prehistoric population and perspective for the Dougga site. Okay. Does it work? Okay. So my presentation will have this plan. I will start with an introduction to introduce the prehistoric background and the historic background of North Africa, then the historic of ancient DNA and some applications. The second part is about our results on the investigation of ancient DNA in North Africa, mainly in prehistoric populations. And the last part is about the ancient DNA study proposition for the site of Dougga. So let's start. As you can see in this map, North Africa has a very interesting geographic position. It's in crossroads of Europe, Sub-Saharan African, and Near East. And this very interesting position led to a very civilization and migration of human migration that occupied North Africa since the prehistoric time. Indeed, the oldest presence of human in North Africa corresponds to Homo erectus remains discovered in Ternifil in Algeria, and several fossils discovered in Morocco highlighted the transition between Homo erectus towards archaic form of Homo sapiens. And the Homo sapiens sapiens, the modern human, appeared in North Africa with the Archean industry, with human authors of the Archean industry. In the Epipaleolithic period, this is a period between 23,000 years to 10,000 years before present, Meshta el-Arbi man appeared in North Africa, and this man is the author of Iberomorusian civilization. This is the civilization of, this is the Iberomorusian civilization. And as you can see in this site, in red spot, the Iberomorusian sites are mainly located in the littoral of North Africa, and the most important sites are Tafouralt in Morocco, Kolumnata and Afalou in Algeria. And after, in the Mesolithic period, the Capsian civilization appeared in central and southern Tunisia. This is with the Capsian man and Capsian art. And after this prehistoric period, we have the historic period. And as you know, for the historic period, we start with Berbers, called also Imazigh, which seem to have Capsian as local ancestors. And after, they are Phoenicians, Romans, Vandals, Byzantine, and Arabs. So, as you can see, we have a very rich background at prehistoric time and also at historic time. So the knowledge of the North African settlements can be studied and established with two approaches. The first one, the study of archaeological specimens and their environments. The second, the study of genetic diversity of human populations. And now, allow me to present the ancient DNA discovery and applications. So, the germination of groundnut seed, aged of 550 years, is the first experiments highlighted that genetic materials can be preserved in ancient specimens. And after, in 1984, Higuchi and collaborators isolated isolated DNA from a desiccated muscle fragment of the quagga. And this is the photo of the quagga. The quagga is a species close to the current zebra, but disappeared since 1883. So this is also another application of ancient DNA at that time. And 1 year after, Pebo, very famous researcher in ancient DNA, obtains for the first time DNA fragment extracted from a dried muscle of 2,430 years old Egyptian mummy. And in 1986, DNA isolated from human brain in Florida highlighted that DNA can be also preserved in soft tissue and not only in dried tissues. The improvement of molecular biology technicals, and in particular the discovery of PCR. Now, PCR is very famous with the COVID-19 epidemic. PCR polymerase chain reaction. With this reaction, we can amplify DNA. So all this technology gave a dash of for the ancient DNA field. So ancient DNA now is can be easily isolated from several materials such as bones, animal and plants, desiccated tissue, hair, teeth, soft tissue, and even coprolites. And we can also extract and isolate DNA from very old specimens like Neanderthal and mammoth. So, after this introduction regarding the background of North Africa and the history and the application of ancient DNA, I have the pleasure to present you some of our results on the investigation of ancient DNA in prehistoric population of North Africa. And our aim was to better understand the origin of North African settlements by the study of mitochondrial DNA diversity of Iberomorusian populations, skeleton from the archaeological site of Tafouralt in Morocco and of Afalou in Algeria. So allow me first to tell you that in human we have two types of DNA. We have a DNA located in the nucleus. Here, this is the cell. The nucleus, here we have the DNA represented by chromosome, and this DNA is inherited from mother and father to the child. And there is another DNA located here in this organite called mitochondria in cytoplasm. And this DNA is mitochondrial DNA. It's circular DNA. And it is only inherited by the mother. Only the mother gave her mitochondrial DNA to the child, daughter and son. So, mitochondrial DNA is an important tool for human population genetics. In fact, it's a circular DNA. It's a small DNA compared to the nuclear DNA. It has its maternal inheritance, so their absence of recombination, and high mutation rate compared to nuclear DNA. So, thanks to this characteristic in mitochondrial DNA, we can follow the migration of population and we can follow the generation and found origins. And I will tell more in this slide. Here, mitochondrial DNA variations can be classified in different groups that we call it haplogroups. And these haplogroups are specific to continents. For example, in Africa, we found lineage L1, L2, L3. So if you found an individual with mitochondrial DNA L1, for example, we can say that the origin of this individual is Africa. If others, A, B, C, D, is specific to the American continent. If you found an individual with A, for example, haplogroup, so we can say that his origins is in the American continent. In North Africa, we have U6, which is specific to North Africa. And in the Mediterranean region, we can find T, U, V, W. So we shared several haplogroups, in the Mediterranean region. And here also, another representation of haplogroup. And this analysis, we call it phylogenetic analysis. So you can see here Africa, and we have the datation of the haplogroup. So this analysis showed that African population are the most ancient population in the world, in the humanity. So African population are the most ancient. And after that, they are the out of Africa theory. So the migration from Africa to the settlement of Europe and Near East and American continents. So here we have also L dated of 2000 years. There are other old African haplogroup like M1 here and U6 other Africa. U6 is specific to North Africa, is around 55,000 years. Okay, I said that we chose to work on Iberomorusian. Why this shows? Because the origin of Iberomorusian men in North Africa is subject of great debates. There are 4 propositions and 4 hypotheses. They can have an European origin, a Near East origin, Sub-Saharan origin, or North African origin. So we studied Taforalt in Morocco. Taforalt site is a cave located at 55 kilometers in the northwest of Oujda in Morocco. And this is very famous cave because they allowed the discovery of more than 200 skeletons. And our work was performed on 30 samples. The second site is the population of Afalou, is the site of Afalou in AGYA. It's situated at 30 kilometers from the current AGYA in the northeast of Algeria. And this rock shelter delivered 50 individuals. But unfortunately, in the collection of Institut de Paléontologie Humaine in Paris, there are only 5 individuals. So here are our methods. So to work on ancient DNA, we need to take drastic precautions to avoid contamination with current DNA. So we work in a clean room, what we call the clean room, and we have to wear gloves, mask, to protect the specimens from our DNA. And then ancient DNA was isolated from bones, and we amplified with PCR the control region of mitochondrial DNA, and we directly sequence it. So you are, I think now you know what is the sequence. We sequenced COVID-19 virus, so now it's very popular to have sequenced. So we obtained these mitochondrial DNA sequences that we compared to a reference. And they are a reference for mitochondrial DNA. And this alignment, what we call the alignment, this comparison, allow the identification of variants, genetic variants that are specific and that characterize the individual. And after that, we can classify the individual in the group. Okay, here, for example, this sequence can be classified in the haplogroup T. And of course, we can do the same work for the other individuals and we can classify them. Also, we can perform phylogenetic tree and network, and I will show you some results of this analysis. So here, the results of the genetic structure of Taforalt in Morocco. And as you can see, the individuals are classified in haplogroup H, GT, and U6. And these haplogroups are originated from Eurasia, regarding H, U, GT, and V, and U6, as I said before, U6 is specific to North Africa, so they are a component, a North African component in this population. we notice the absence of sub-Saharan haplogroups, the L. We didn't find L haplogroups here. This, and it is important to note also that GT haplogroup nowadays is only observed in Berbers from northern Morocco and in Sicilians and in other Italians. Then, this is the results of genetic structure of Afalo specimen. And as you can see here, we have only Eurasiatic components. We didn't find also sub-Saharan haplogroups. So, based on these results, we can exclude the sub-Saharan origins of Iberomorusian population, Taforalt and Afalo, because we didn't find this lineage. We find European origins, Near East lineage, and North African. So we can preserve these three origins, but the sub-Saharan is excluded. And this is our work published in 2005. So, when we compare now this genetic structure of prehistoric population with current population in North Africa, and as you can see here, the current population of North Africa has three components. North African component represented by the U6, Eurasiatic component, and sub-Saharan component. Nowadays, current population of North Africa has this sub-Saharan component that is absent in the Iberomorusian populations. So here another examples. As you can see, North Africa, I represented the three lineages. In blue, European lineage, in red, sub-Saharan lineage, and in green, North African lineage. These are North Africa, they are Near Eastern population and European population. And as you can see, the main component in European population are European lineage in blue. And in North Africa, is very specific. North Africa is very rich. They are the three components. And so we can say that our genetic structure in North Africa is like a mosaic. We have components from Europe, from Sub-Saharan Africa, and from Near East. It's very, very interesting. So here, thanks to our results on prehistoric population, we can precise the chronology of the appearance of Sub-Saharan lineage in North Africa, here represented in green in the spots. So we propose that Sub-Saharan lineage, the Sub-Saharan migration to North Africa are dated after 13,000 years, because we didn't find this lineage in Afalo or in Taforalt population. This is what we called multidimensional scaling analysis. This analysis allows to compare populations, and it's based on the genetic distance calculated from the sequences of mitochondrial DNA. So you can see here Taforalt, Taf, and Afalo in red, clusterized and grouped with the population with the Berber population in Morocco and in other population in the Mediterranean, such as Valencia in Spain and other islands and other Spanish islands like Minorca and Mallorca. So we can say that there is here an existence of genetic flow, genetic migration between southern and northern coast of Mediterranean. Here with we can see their migration between the Spain, between Mallorca, Minorca, Sardinia, and Taforalt, and also a genetic continuity in North Africa since the Epipaleolithic period. Do you remember the haplogroup GT? It's only observed in Berber, in Moroccan Berber, in the same locality of Taforalt. So there is genetic continuity since the Epipaleolithic period in this locality. So let's move now to the last part of our talk. It's about our proposition, what we can do for the site of Dougga. And this is the new project. We thank AGYA for supporting these projects, and which a project in collaboration with the Institut National du Patrimoine and Institut Pasteur in Tunis. As you know, and now you know it because Fatma too showed this morning, and Samir Aounallah also, that there are unanswered question regarding the site of Dougga, mainly question related to the biologic aspect of the population of Dougga. So, archaeological investigation have illustrated the cohabitation of Numidian, Punic, Greek, and Roman cultures on the antique site of Dougga. So the question, what is the impact of this cohabitation on the genetic composition of the population? Another question. Recent excavation on funerary monuments have identified heterogeneous disconnected human bone masses. This is the work of Fatma Touche that showed you the burials with lots of bones. And the question, is it a family burials? And due to the bone fragmentation, conventional anthropological or archaeological methods cannot be applied to determine the relationship between the buried humans. So here, ancient DNA can contribute to resolve these questions. As I showed in the second part of the work, DNA can provide information about ancestry, ethnicity, the relationship between ancient population that cannot be accessed by other approaches. All these activities can help to better understand the human past. So, for our project, for Dougga, this is what we proposed. DNA will be isolated from petrous bones, here the photo, and teeth of around 50 samples. We will work on about 50 samples. And first, we will determine the genetic fingerprint of samples by studying nuclear DNA and mitochondrial DNA. So we can, thanks to this study, we can characterize the individuals inhumed in the same burials. We can determine the sex, the genetic profile, and also we can deduce the relationship between them. So we can deduce and we can conclude if the burial is a family burial or no. The second activity is the analysis of mitochondrial DNA lineage of samples in order to identify the genetic composition of the antique population. So here, if we identify the haplogroups, the mitochondrial DNA haplogroups, so we can determine the origin of the population. Remember, the L haplogroup L is originated from Africa, U6 specific to North Africa, and the G, U, T, etc. We can find it in the Mediterranean region. The third activity is the analysis of genetic variants in nuclear DNA, mainly variants associated with the phenotypic traits, such as the color of the skin, the color of eyes and hair. So we can establish a phenotype characterization like a portrait robot. You can see if the population of Dougga have the green eyes or brown eyes or other. So we can imagine, and you can have for samples some characteristic to know more about this population. And the fourth is regarding disease. So we can study also genetic variants involved in disease, like common disease, metabolic disease, diabetes, for example, or cancers. So we can identify some targeted diseases, and we can know better the population and what kind of disease that involved in this population. All these results associated with other specialties in archaeology can help us to deepen our knowledge on the population of Dougga. Thank you very much for your attention. If you have a question, I'm happy to answer it.
for the questions.
You have a question?
No, I have a question, yes.
Okay.
But the floor, please. Audience. Any questions? Okay, I think you are looking for DNA with good quality, not great DNA. What about the nuclear DNA in terms of conservation? Is it better conserved than the mitochondrial one?
Okay. Mitochondrial DNA is better conserved than nuclear DNA. Okay, because it is circular, it is small, and it's found in a very number of copies in the cell compared to the nuclear DNA. But nowadays, there are progress in molecular biological techniques. So, you know, next generation sequencing, and we have a lot of new materials and new extraction kits that allow to have a very good quality of DNA extracted, even from fragmented bones or fragmented tissues. So, you know, even the nuclear DNA is less conserved than mitochondrial DNA, but thanks to these new techniques, we can have results. Additional information, because it's important. Because regarding the variants involved in disease, they are nuclear variants. Regarding variants involved in phenotypic traits, are also nuclear variants. So DNA, mitochondrial DNA, is not enough to have a lot of information regarding the population.
Good. Yes, sir.
Yeah, it's much fascinating for me to listen to. I was wondering if I also can say something about possible origin. I think you can also say something about timings, like when
the time.
Yeah, the time when approximately people would have been maybe in one place and then they arrived in Dougga. Can you tell us on the DNA?
Of course. I come back to my slides here with the phylogeny, you remember? And they are datation, yes, the time scale here. You have the time scale. So here in Africa, we know that L1, L2, we can find that 100,000 years, and the other very recent haplogroup like H, 20,000 years, or others, V, HV, very, very recent. So we can estimate the origins of the population and the time of the genetic flow, the time of the migration. Yes, for example, when we found in Dougga, for example, I don't know what we can find it, perhaps U6, because it's originated from North Africa, and U6 is around 55,000 years. So we can say that their continuity in situ in North Africa from Assyrian, from the other period, prehistoric period, in the Roman burial or others. Is it clear, Barbara? Yes. We love you.
Thanks so much. I have an idea about the relationship between evolution, progression, and fashion.
Okay. Yes, very good question. Okay, here I come back to the slide regarding the origin of North Africa, of Iberomorusian. Okay, so as we know that Assyrian preceded Iberomorusian culture, but European origin for this North African origin, if we, so we found U6. So U6 is North African origin. So we can say that North Iberomorusian, some individuals from Iberomorusian populations are originated from North Africa, originated in North Africa. So and what population before Iberomorusian are Assyrian? So we can say that their genetic continuity in situ in North Africa from the Assyrian to the Iberomorusian. But we need to know also the genetic background of Assyrian. Perhaps the Assyrian we can find also U6, and you can find T and other Eurasian haplogroups. Okay, so if you have sample from Assyrian, I'm happy to study it.
caption population, the caption one.
Yes.
There is a relation between the caption one and the not the
So the captions is after Iberomorusian.
Yeah.
Okay. So also you have to study the caption population to know.
You have no idea about
No, there are no study on this population. I remember that when I started working, I don't know. Yes, you have skeleton, but you need to have a budget. You need to have a budget because it's very expensive. You know, the reactive reagents used for molecular biology is very, very expensive, and you need to have a budget dedicated for this. And fortunately, for the project of Dougga now, for these samples, these 50 samples, we have the support of AGYA. For this project, and so we can study it. And I hope that with you, with other colleagues from Institut National du Patrimoine, we can submit other projects for in the frame of international collaboration, for example, like Horizon Europe or other grant office that we can help to, yes, to have money and to study.
Working on science from Tunisia can be
collaboration
with you.
Of course, I'm open to all of these collaborations. Yes.
Sorry for my stupid question, but the caption is prehistoric population?
Neolithic is after.
So what about the DNA? You can envisage that the DNA will be very degraded? Or...
Okay, this is a good question also, Alpha, because here DNA is not really related to the age of the specimen, but exactly, it is related to the environment. Okay, we can have, I worked on the specimen of Tafurat, and I worked also in a specimen of medieval population in France for my master degree. And I found that for some samples of Tafurat, we have a good quality of DNA, and for other very, very recent, but we didn't find any amount of DNA. So it's linked mainly with the environment and the mode of conservation than the time and the age of the specimen. Other questions?
Maybe I have a particular philosophical question. Because if we agree on the fact that the mankind was born in Africa, or should we look for the origins of the North African population out of Africa?
Out of Africa? Yes.
There is no reason. Everything. I mean, if all the mankind was originated from Africa, why are we looking for the origins out of Africa?
Okay, I understand.
Population.
I understand your question. So, but the origin in Africa is the first origin, but now we are different. We accumulate a lot of variation and mutation. So L haplogroup is originated of Africa, but in our population in North Africa or Tunisian, we don't have only L. We have H, we have J, we have K, we have, you know. So they are originated from other regions from North Africa. I explain more. The most ancient sequence, mitochondrial sequence, are originated in Africa, but after they are evolution and there are other sequences, you know. So it is important to study each population in the world and not only Africa, because they are migrations, as you can see here. So it's clear. Thank you. We did.
So thanks for the presentation. It is always a pleasure to assist at your conference and process. My first question is, what is the reference sequence used in general to detect the haplogroups and determine the genetic origin of individuals?
Okay. Yes, very good question. Yeah.
Especially that you said that the first occupation in the world.
Yeah, of course. The reference sequence, it was established in 1984, I think, and it was like a consensus reference. So they sequenced some people from Africa, some people from Europe, some people from Eurasia, okay? And then they performed phylogenetic analysis. And when they have the phylogenetic analysis, they demonstrated that the individual originated from Africa are in the bottom of the tree, phylogenetic tree, and that conclude that it's very ancient. But the reference sequence is what we call CRS, Cambridge Reference Sequences. It was for the first time discovered in Cambridge University by Anderson. And for this, it's now this Cambridge reference is equal to the haplogroup H, the first sequence of haplogroup H, the most simple one. This is a consensual sequence. Okay.
What methodological strategy are we going to proceed in this project?
Okay, so our strategy here is, first of all, our contribution to this project is to help to resolve these questions. This question related to the biological aspect of the population. As we presented, me and my colleague in this morning, we can't determine the sex and the age and other information about samples. Okay, so our budgets can be used only to have 50 samples. Okay, so we will start with these 50 samples.
Which is huge.
Yes, of course, because in my thesis, for example, in my PhD, I worked only on 30 samples from Tafuralt and 7 samples from Afalou. Yes.
That's very close to the genome.
Exactly.
15,000 euro per genome, which is for the current DNA costs about 1,000 euro.
Exactly. And we will perform next generation sequencing with also very expensive compared to only one fragment with PCR and Sanger sequencing, you know. So our strategy is to extract DNA with good quality, to perform next generation sequencing for to sequence all mitochondrial DNA, to determine haplogroups, and to determine the relationship. So we will use nuclear DNA for the relationship between individuals, mainly those inhumed in the same period. And we can also, with other samples, so to know the genetic background of the population, and we can deduce the origins, variants, as I said, the phenotypic variants, and variants involved in diseases and others. This is our strategy. Okay, so we are driven by the question of the archaeologist and the anthropologist of the site.
Thanks for the response and answer. And the question is a bit. So, what do you think about North African Asian human sequence? So, it is possible for us to, just like the human reference, we have
Okay.
And it is an Asian sequence, so that we can compare it to another sequence of us, and to determine, first of all, the histological background, and to determine the timeline of the evolution of diseases, especially the frequent diseases, and determine especially the contribution of the environments in our zone.
Very good comments. And I didn't think before to have a reference specific to us, because the reference is now established. But it's very important to have, I have to say, it's amazing to have these young people with this fresh idea, new idea. We are working with a reference. Why you use this reference? We can have our specific reference. Yes, of course. Exactly. I agree with you.
This is why we are working on local databases and trying to identify variants and compare them to the variants identified in our
Exactly.
Because it's meaningless to find a variant in an African and compare it to European. It can be benign here, it can have phenotypic or any other involvement in other population. It depends on the whole context, genetic and environment. Which is raising a good point.
Yes.
You remember, in the meeting with our partners from Max Planck, Johannes, they said it's very nicely project. We will look for variants, we will bring this software robot. It's nice. But practically, things are not going to be so easy. Because if we are looking for variants for local individuals that are responsible of the blue eyes, etc., we cannot extrapolate findings from this population to other populations. We have to standardize the populations according to what is observed at that time, then we can conclude. So it will be biased a little bit, but we don't have a choice because
we don't have a collection of
yes. Working like for the police, scientific police, it's possible to do these parameters to fix them based on the investigation of local and individuals. It's okay. Now we have the parameters. We can tell the software that if you find this one, it's a blue, it's etc. You have this distance between eyes. But if you want to extrapolate these parameters to ancient population, I am sure you will find
some variability.
Many biases. And the second point, we are working with findings on populations local to a site which is like Samir said, it's a mix with Punic, Roman, and etc.
Greeks and others.
But we have no choice. We have to do, and then maybe we have to change the parameters to do it.
Yeah, to adjust one comment, we are working now on the traits involved in colors of skin with Haifa and Durra Bishr from the police scientific to know what kind of variants involved in this trait in our country. But we start with Mediterranean populations, and after we can use our data to explore these results on Tunisian population, for example. So it is important with it. Yes, very, very important question, but we need to start because we don't have database for ancient population in North Africa. We have databases with Neanderthal, with Cro-Magnon, with other Europeans and African fossils, but we have not enough information about our population, and we start working on this, and of course,
good luck for us.
Yes, exactly. Thank you very much.
Okay, and now it is my great pleasure to introduce our next speaker, my wonderful colleague and fellow AGYA member, Nadia Bahra, who comes to us from Algeria, from Université de Constantin, who will talk to us, who will actually continue this fascinating discussion about ancient DNA, archaeological evidence, and will talk to us about just this topic and draw on her own examples from prehistory. We're really looking forward to your presentation.
Oh,
Yes.
yes. Thank you. Thank you, Barbara. Thank you, Olfa. Thank you to all, to AGYA members and colleagues from ENP for inviting me to this workshop. So my presentation or my contribution here is not specifically about Dougga or the Roman times. It is about how ancient DNA studies were seen by archaeologists at that time as a fresh boost for many archaeological issues and in an interesting way to reflect on old questions with new tools. As you know, ancient DNA studies occurred first on prehistoric samples and was seen by prehistorians like me as a way to overcome the lack of written sources before the fourth millennium BC. Their impact is much greater in studying prehistoric society than the antique one, as much as was the absolute dating revolution in the 50s. So we have many revolutions in archaeological research, and the last revolution was dating, absolute dating, and now we have a revolution with ancient DNA analysis. At that time, there was no chronological frame other than stratigraphy, and suddenly most prehistoric culture got an approximative age. So now we have the same revolution. We have a lot of issues that have no answer, and now we hope that we'll get new answers for this problem. And the main issue that we can deal with the ancient DNA is the issue of diffusionism or migrationism. So it is cultural diffusion or migration of population. It is a very interesting topic in archaeology, and for many times we don't have answers, and now I think we can have more information about this. Okay, so before the 80s, origin and filiation of animal and human species were investigated by the means of paleontology and physical anthropology. However, the conclusions were sometimes subjected to debate and disagreement among academics. But by the second half of the 18th, first ancient DNA sequences were published, and paleogenetics seemed to offer a way to confirm or deny the results of morphological phylogeny. The first sequence was from an Egyptian mummy. So yes, you can see here, which was dated from more than 2,400 years old. And the second sequence was from stuffed quagga. It is a zebra-like animal extant in the end of the 19th century. And these first attempts at that time were welcomed by archaeologists with a great enthusiasm. And it appeared after that there were a lot of problems in the sequencing, but at that time it was really a revolution. After between in the 90s, yes. In the 90s, there were a dozen of publications that were issued announcing spectacular results on ancient DNA extraction.
We have plants. So DNA, chloroplast DNA sequence from plants. We have also Mesozoic insects, so DNA sequences from a Mesozoic insect. It was draped in amber, yes. So this insect was draped in amber. We have also a sequence from a dinosaurs, but unfortunately, after new studies, so it appeared all these results were proven wrong, and paleogeneticists understood that we have to be careful in our extraction, cloning, sequencing, and we can't just came and extract ancient DNA and sequence it. So from the side of archaeologists, there was a climate of such suspicion. Okay, so when they see that these results were wrong, so archaeologists say, oh no, we don't need this technique. The results are not serious, so we can't use it. It was exactly like carbon 14, radiocarbon in the 60s, where some results didn't fit archaeological results, and there was a little skepticism among the specialists. But after the lesson taught from these results was that extracting and replicating ancient DNA is more complex. It's a complex process that needs careful manipulation and strong understanding of limitation of the techniques. To understand, I know that my colleague Rim Kefi gave a wonderful presentation on the subject, but I just try to give an insight from my point of view. So we have several techniques, and the most now is PCR, so polymerase.
Chain reaction.
Voilà, polymerase chain reaction, and sorry, yes, I am. Yes, what are the limitations? I have to tell the limitation first. So there are many technical limitations that are specific to the field of ancient DNA. We have the poor preservation of endogenous DNA, and the presence of contaminant exogenous DNA. So we have to know what is endogenous and what is the result of contamination in the excavation, in the laboratory, and so on. So the difficulty is that the consequence of degradation process can cause miscoding lesion, sequence error, physical destruction of the DNA molecule, and this increases the risk for preferential amplification of exogenous contaminant sequences. So when you have this problem, it's difficult to produce sufficient quantities of authentic DNA to make the study conclusive. So there are several red flags. There were several red flags at that time, and now paleogeneticists are aware of all these problems, and they take care when they work on ancient DNA. The methods include several steps: extracting, amplifying, cloning, and sequencing. The classical method is, as I say, polymerase chain reaction and Sanger sequencing. The method used in the 90s, at that time, it allowed only to obtain short DNA sequences, and the results were deeply impacted by contamination. But after, in 2006 and 2007, there were several improvements that were suggested, like multiplex PCR that allowed to get longer sequences. We have also specs amplification and other techniques. So I have the paper here. Yes, well, it's a paper from Johannes Kroos, and we have also a large-scale sequencing of mammoth DNA. And now, today, we have a new methodology that is expensive, but give better results. It's next generation sequences that was explained by my colleague. And this method increases the number of basis sequences per run. And also, this technique gives more accurate results. Addressing the limitation of addressing the limitations of ancient DNA studies requires a close collaboration between paleogeneticists and archaeologists. We have to avoid contaminations and collect skeletal materials with we have to manipulate only with gloves and face masks during excavation. We have to select archaeological remains for which the taphonomic history is well known. We have to avoid washing samples and not stock them in plastic if we can. So all these actions are done by archaeologists during excavation. So if we want to do DNA analysis, we have to avoid all these practices. The ancient DNA can also be deteriorated by variation of temperature, pH, humidity, several factors. And it's for that, so we have to put them in, to store them in a cool place. And the problem here is with the ancient or old collections that are not stored in the correct way and were not extracted in the correct way. So what are the information that we can get from human ancient DNA? We have the main result is to validate and discuss the results of paleoanthropological studies. So it can give us information on the relationship between individuals, the genetic distances with other populations, the interbreeding or migration, and sometimes societal organizations. But now most examples for us came from Europe, because in North Africa, we have only a limited number of studies, as Rim explained it to us. Okay, at the beginning, one of the most known and surprising results about human ancient DNA analysis was the result of DNA sequencing of Neanderthal. And at that time, no one among archaeologists can imagine that this result can happen. We always think that there wasn't any interaction between Neanderthal and Homo sapiens or anatomically modern humans. But now there are these results that show possibility of interbreeding in Europe, at least in Europe. And studies also show that the Neanderthal genomic sequences represent 1 to 3% of the genome of modern Europeans, and that these sequences are expressed in the phenotype, especially in the immune system response to some viruses. And we all know the case of the response to COVID-19, that as there were more complications, several complications due to the COVID-19 in Europe than in Africa. In Asia, we have also the discovery of an archaic hominin, Denisova, that wasn't known before. And why it wasn't known before? Because the human remains of Denisova cave were not very characteristic morphologically and display a mosaic of character of anatomically modern human and Homo erectus. But the DNA showed that there is something more complex than what we think. They also find that the Denisovan genetic material contributed by 4 to 6% to the genome of present-day Melanesian and population of South Africa. But the contribution of DNA analysis to prehistoric archaeology is not only about archaic humans species, but it also deals with one of the oldest issues in archaeology: are cultural changes related to human migrations or to transcultural diffusion of ideas, the diffusionism. That was summarized in one sentence by archaeologists. It was pots, not people. This discussion is particularly illustrated by the question of the origin of Neolithic in Europe. There was a discussion between the partisans of a cultural diffusion of the Neolithic in Europe by the Middle East and the partisans of population migration. And recently, this discussion was settled by ancient DNA analysis, which showed the role or farmers' migration from Anatolia, Turkey, in introducing elements of the Neolithic culture in Europe. In addition to domesticated animals and plants, circa 8,500. These Oriental farmers mixed with Mesolithic hunter-gatherers in Europe, and we have a lot of results about this. So it's really a revolution for us. For the first time, we know that Neolithic wasn't a migration in all the parts of the planet. We have another example, is the origin of the Bell Beaker culture in Europe, la culture campaniforme. So maybe I have a photo of Bell Beakers. Yes, okay. So this culture is characterized by inverted bell beaker drinking vessel and palmella points. And it's characteristic of the beginning of Bronze Age in Europe. You can find also elements of bell beaker culture in North Africa, in Morocco, and in the west of Algeria. So the question is, the bell beaker culture the result of migration or cultural diffusion between Western and Central Europe between 2800 and 1800 BC. The DNA sequencing was performed on the remains on 400 individuals from Neolithic to the Bronze Age, and it showed that bell beaker culture, bell beaker population were different between Central Europe and Southwest Europe. The first ones related to migrants from the steppes who arrived in Europe at the end of the 4th millennium. The seconds are the descendants of Anatolian Neolithic farmers. So the result is clear. The material culture of the bell beaker diffused regardless of migration, and there is only one exception, the UK, where the bell beaker culture is associated with the replacement of 90% of the genetic pool. So we can ask the same question now about the bell beaker culture in Morocco. Is it migration or a cultural diffusion? But we have no data. There are a lot of examples. I won't cite them all, but to summarize the other contribution of ancient DNA analysis combined with archaeological data is the understanding of prehistoric societies, like the revelation of female exogamy in the elite of South Germany in the Bronze Age. It's a study from 2019. We are also the possibility to know the phenotype of some characters, like the apparition of blue eyes in the Mesolithic and so on. There are of course, limitations in that, that we need to be sure that the mutation is actually linked to the phenotype by comparing with modern population, and this is difficult for extant species. Before this, I will not talk about North Africa because my colleague have given a presentation on this. I will only say that we have the result of Rimkefi that says that it's possibility of continuity between Arthurian and Iberomorusian. So as an archaeologist, I can be surprised because we don't have a smooth transition, cultural transition between Arthurian and Iberomorusian, and rather we have a hiatus in the sites, stratigraphic and cultural hiatus. Thus, it's a big question for us, and we hope that we will find an answer. There is also new results, recent results with next generation sequencing that showed for the Neolithic in Morocco that there is no evidence of migration at the ancient Neolithic. So the beginning of Neolithic is from local people, but after there are elements from Spain in the recent Neolithic, and also from the Beaker culture. So it's also a result that is interesting. Contribution of DNA is not only about humans. We have also results from animals, and it can inform us about their migration, extension, expansion, replacements. There are a lot of studies about wild species, but the most interesting for archaeologists is exploring the origin of a process of domestication in the beginning of the Neolithic. So I will give only a few examples. As we know that the early phases of animal domestications has been eluded by biologists and archaeologists for decades, and from species that were domesticated from geographically restricted wild ancestor, things were clear for us. But there are species who don't have this information. So the DNA study would be very useful for us. We have the time, please, just to know. Okay, I continue. Okay. It's good, I can. For the study of domestication, we have two approaches, two complementary approaches. So the first approach is based on the archaeological record, documents morpho-anatomical changes and cultural innovation through space and time. We have to seek for markers who show the shift in the relationship between human and animals. It's indicators of domestication. We have, but this record is partial, fragmentary, and it doesn't give us informations like coloration, fecundity of domestic animals, and all this. So it is the most important information in the early stages of domestication, but unfortunately, archaeology can't answer us on this. The second approach is genetic data obtained from modern domestic animals who can retrace their geographic and temporal origins. But we have also the approach of ancient DNA that enable access to genetic information from past animal population, offering the opportunity to combine the time depth of archaeology with the resolution of genetic data. We have the example of domestication of goats. For which the distribution of their wild ancestor was geographically far more limited to Near East. But ancient genomes have revealed the complex contribution of multiple genetically divergent wild lineage to early domestic population. So the genes selected during the domestication process are involved in coat color, fertility rate, and milk production. The pattern for goats, domestication of goats could be the result of a continuous incorporation of wild individuals into the domestic population, or an independent process of domestication in different regional settings. This is consistent with the archaeozoological evidence that show the domestication of goats in the Near East was dispersed in time and space. We have also the example of domestication of pig. So the pattern is more complex here. Pigs were domesticated from Anatolian boars, were introduced to Europe by Neolithic farmers. So the domestication of pig is not local in Europe. But after the domesticated pigs from Near East were mixed with locally present, with locally European boars, and we can see that the genomic affinity of the first domestic pigs transported into Europe was nearly completely erased by gene flow with the European wild boar, except for their black coat color. And after DNA studies shows that these mixed pigs were then, or interbred pigs, were then reintroduced to the Near East. So it's very complex information that maybe we cannot understand with only archaeological data. There are examples about domestication that wasn't resolved nor by archaeological and nor by genetics. We don't have answer till now, like the domestication of dog. Because the reference data from extinct Pleistocene wild canids is absent. And we don't have material culture accompanying early stage of domestication of dogs. So the analysis of ancient dog genomes and the archaeological pattern of dog suggests that modern dog population may be derived from independent wolf population in Western and Eastern Eurasia, but this interpretation is still questioned. And the last example of domestication pattern that is still unclear despite ancient DNA analysis is the domestication of horses. We have the morphological changes that differentiate between wild and domesticated horses only appear in the early Iron Age. But the earliest evidence of harnessing, milking can be found before in 5,000 years ago in Central Asia. So we have several archaeological hypotheses that are partly contradicted by DNA data, showing that horses from Central Asia are not the ancestors of modern horse. but they could be clones of other type of horse, and there are still a lot of questions about this. Last thing, I will not speak about results from plants because they are limited. We are only one example about corn domestication in Central America, and it's not, there are still a lot of questions. But there is also a thing that we can do with ancient DNA, other possibilities like identifying animal species on fragments that cannot be identified by paleontology. We can also have evidence of human DNA in sediments of sites where there are no bones. So it's an evidence that humans were there, but we have no bones, only DNA on sediments. As a conclusion, the contribution of paleogenomics to archaeology are multiple and important for all chronological periods. We need the close collaboration between archaeologists, bioarchaeologists, paleogeneticists. It's necessary throughout the study process, from the field to the storage of collection with the establishment of strict protocols for sampling and conservation. It's also necessary to agree on the hypotheses to be tested by a paleogenomic approach. This is about archaeological hypotheses. Finally, it's fundamental to compare the data from the different experimental approach used and to work together in the interpretation phase of the results. Thank you.
Let's open the floor for questions.
And maybe I can.
I have a question. Thank you, Lydia, for the presentation. Do you know if now in Nigeria there are teams working with ancient DNA in a second area to another institution?
It's not a team, but it's an idea. We have people who are interested in this, and maybe we will think about making a team and maybe connect with other colleagues from Tunisia, Spain, or other countries. So it's the idea was discussed, but practically not.
Okay.
Yes.
But there are people.
Yes, biologists.
Biologists.
Yes,
Okay.
with biologist background.
Okay.
Yes.
Nice.
It's the same.
I was saying what exactly about the results. What were the results? Did they explore the mammoth in terms of structure in chromosomes, which specific fields, or they just investigated that parts of the what were the results?
I will give only the archaeological answer, and Rim will give you.
I give you the answer. There are several, I think, more than 2 works on mammoths. There one, I think the second or the third work was contamination from the mammoth. But there are another work when the mammoth is conserved in ice. Very well observed. So, thanks to this work, they know the phylogeny of the mammoth and the position of mammoth in the phylogeny compared to elephants. Elephants from Africa, elephants from Asia, etc. So, thanks to this work, we know the position of these species and the dates of the separation between elephant and mammoth.
Yes. It closed a debate among paleontologists for a long time, and it appeared at the last that the first paleontologists were right. Okay. Another question.
Well, if not, I think
Oh.
thank you very much
Yes.
for these insights. And I wanted to, it's very brief. I really enjoyed that you took us almost into the history of the discipline of archaeology, how you got close to genetics and then got
Yes.
skeptical. I found this super fascinating because you can never anticipate what will happen next in terms of innovation. And when you talked about museums, I was wondering, is there any way for the museums today, for the stuff they have in the collections that is maybe contaminated, can they do something to still use it for these kind of?
I think it's a matter of change. We can find and we can not find. Okay, it's museum collection. There is a problem.
Museum collection. Yes, I worked on museum collection from IPH, Institute of Biology in Paris, for the collection of Thugga and Beaker. Yes. I agree with you that where these samples are very contaminated because they are captured with
They were washed.
Because since the excavation to the museum, but now we have a technical how to say drop to the surface the superficial surface of bone, for example, and we take the inside of the bone. So we can work on this.
So not all is lost. You can still go to
Yes.
samples and
Yes, to go inside of the samples and to have other. And of course, when we found teeth, because teeth is very preserved, and the dental DNA in the teeth are inside. So the outside is the mineral. So it can protect the DNA material. So we can have a section, longitudinal or transversal section of the teeth and of teeth, and we can found probably the DNA inside.
I don't know if I am right, but when you worked on the Arambourg collection, the IPH, you didn't find DNA in all the samples.
Yes,
Yeah.
yes. I remember that there are only 8 long bone, and I found only in 7.
Yes.
Because one is very different.
Thank you so much. I mean, this already shows the panel was really interesting because it brings
It's
you
complementary.
even
Yeah.
in the question section. So it is my pleasure now to complete this and introduce the last speaker for today. And our last speaker is Dr. Beaker, who's also an academic joining us from Algeria today. And she will speak on her research on Dougga, and she will analyze architectural and urban specificities in this region for us. And
thank you. Thank you very much. I'm Dr. Lydia Mahloufi. I'm from Algeria, and I'm a member also. And I'm teacher at the School of Architecture and Urbanism in Algeria. So I'm going to show you a work that I did on the south of Algeria and the adaptation of the housing and the habitat to the environment and the surrounding environment. So it will be different from the previous presentations. So here it will be more on architecture and urbanism. So I hope you will enjoy it. Just a second. Okay, so let's start. So I'm going to show you work on Ghardaya. It's in the south of Algeria. I will do an analysis of architectural and urban specificities in Saharan climates. So as you can see here, this is my country. So you have Algeria here, and Algiers is in the north part, and you have Ghardaya in the southern parts of Algeria. So just to give you an idea, so here this is the capital city, and you have Ghardaya. It's we call this the gate of the desert. So this is the first city that we meet when we go to the Saharan part of Algeria. So Ghardaya is a UNESCO World Heritage since 1982, and we call this for the inhabitants Mosabite inhabitants. So this population came from, it was a nomad population, and they settled down in Algeria, and they built 5 cities over there. And the capital of these 5 pentapole is Ghardaya. So as you can see here, I'm going to show you the passive cooling systems, because it's very hot and dry, and they try to adapt themselves and adapt also their settlements to the harsh and dry climates here in Ghardaya. So first I will start by the urban scale. So I will show you the city in general. Then I will show you the building scale. So you have an idea on the city at a whole. So here they took into consideration the local climate and the site context, and they tried to find some strategies to adapt the housing and to adapt also the city to this climate. So first we can see here the microclimate and also the natural treats. As you can see here, so it's we have it's sandy here, and we can see that the winter in general is cold, really cold, and there are also a low rates of rain. Also we can see here an oasis. Not far from the city, and also it's hot and dry summer here, and we can see a big difference between day temperature and night temperature. So it's very hot in the day and during the day, and it's really cold during the night. So here, just to give you an idea, so the microclimate here played a huge role in determining the most favorable building location. And they tried to locate the city just next to the river. So when you try to build a city, a new city, the first requirement is water. So they chose the location in function of the presence of water. So, and here we have the river of the Mzab. So it was the Mzab Valley, and they tried to build their 5 pentapoles near to the river. So this was the first requirement. So as you can see here, we are going to see first the site design and the density. So it's really dense here. So the last census was 311 inhabitants per kilometer square, which is huge. So because it's a small city, so it's a really dense texture. And also you can see here the mosque. So each time you find a mosque, it's one city. So we have 5. Ghardaya is the capital city, and we have Al-Atif. Al-Atif is in the corner, if you want. It's just a translation from Arabic. And you have also Bunura, we have Beizgan, and we have also Milika. So 5 cities, and Ghardaya is the capital city in the Pentapole. So just to show you, you can see here the mosque. So it's the central point, and here you have a small mountain, and on the top of the mountain you have the mosque. So it was used as a watchtower in case an enemy came. So they were just on the top and tried to see the rounds and see if there is a danger or if there is someone coming to the city. You can see here, so just a scheme to show you the city in general. So the lower part is next to the river. So here you will have oasis. You will have also so the market. So it's the economic area. So when a visitor came, or maybe merchants or passerbys, they all enter by the gate, and they are all in the lower part of the city. And the most you upgrade the city, here you will have it's become more private. So, and the religious part is on the top of the city. So you have the mosque on the top, and between the two parts, you have the residential districts. So, and in general, it's like a private part. It's not really open to public. So, and for, you have also the rampart, as you can see, they are really well preserved. Now, just here to show you, sorry. So you have here an idea also about the color, because they respected the color of the sand and the color of earth. So usually the color is adapted to the pigment in the local site. And for the urban texture, you can see here, when you enter the gate, you have on the marble, you have a plan of the city. So for someone who doesn't know the city, who is first comer here, you have a small idea about the city. So you have the souk just next to the gate. Then they just followed the natural site, and on the top you will find the mosque. So you have two main points: the souk and the mosque. The major points here. And for the souk, for instance, it's rectangular. It's the only part of the city when you have a rectangle. So they try to have a regular form, but for the houses, it's irregular forms. So it depends. And also they try to be fair with each other. So you have almost the same surface for each house. So they try to be fair. So you don't have big house and small house. Everyone have almost the same surface. So they try to be fair as a community. For the public spaces, so here you have an idea about the market. So this picture was taken in 2007. So it was my first travel to Ghardaia, and it was the feast of carpet. So they showed a lot of carpets here, and with really good prices. So everyone was coming to Ghardaia only to buy carpet. So it was a local feast. So, and you have here a Mozabite. It's a local inhabitant, and you can see also how he is dressed. So it's also a traditional way of clothes. And you have here, so most of facades were just covered by carpets. So it was really nice for tourists to choose from the external facade and to see directly the products. Okay, so here I can show you also just here to give you an idea about the traditional aspect of the city. And it's the urban spaces. The urban spaces are shaped in a way that they decrease the effect of hot and sandy winds, because we have a lot of winds over there. You can see here also the narrow streets. So as you can see, the site is not flat. So you have to climb, and sometimes they use stairs and sometimes not. So in both cases, the stairs are narrow. So the surface is dedicated to house and to housing. And for the streets, and also for intimacy, because when you have small streets, it's become more private, and also for winds, because as you can see, you don't have straight streets, but you have curved streets. So in case of wind, this decreases the speed of the wind. So they try to adapt themselves really to the climate. And you can see here also, sometimes you have semi-covered streets to have shadows. So here it's just a case when you have a street made of stairs here, and as I said, they are narrow and irregular, sometimes semi-covered. And here, for instance, you can have it's around the market, the supermarket, the souk. So you have here covered galleries. So for all the boutiques around the souk. So it's covered. So for someone who is doing his shopping or her shopping, it's really nice because you are in a fresh area. You can do your shopping quietly and you don't need to rush because it's really hot, or sometimes you can arrive until 84 degrees Celsius. So it's really hard, especially in summer. So to expose little surface for solar radiation in summer and cold climate in winter, the Mosabite have used dense and compact horizontal and vertical patterns. I will show you now the building scale. So you had already an idea about I have already an idea about the urban scale. Now we are going to focus on the building scale. So first, you can see here the facades. So you can see here the prayers are going to the mosque. So it was by 4 p.m. And you can see also the minaret. So the minaret here is not straight. It have also the curve. It's like the car. When you drive fast, you need to have a car which is aerodynamic. So this way you can yeah, you can go faster and you are not stopped by the wind. So in this case also, because you have a lot of wind, it's a windy place. So they try to use curved facades instead of a straight one. So it's, and this way, the minaret will not collapse. Thank you. Okay, so you can see here also the facades. Usually they are almost blind. So you don't find windows, or you have, of course, you have the door, but you have sometimes just a small opening. And this small opening just to have a ventilation. So it's not really, because it's first an Arab Muslim identity. So they try to protect their intimacy. So everything is done inside, and outside we limit the connection between the street and the house. So it's homogeneous system which focus on the patio, and the rooms are looking on the patio. So in both cases, the connection with the outside part of the house is really limited to the door accessibility and also to small openings. So in this sense, it's an Arab Muslim identity, and not specific to Sahara or specific to Mosabite. It's in general in Arab Muslim, also for those who are working on Ottoman period. So you have this specificity. So you can find this typology also in Algiers, for instance, and the Casbah of Algiers, which is also a UNESCO World Heritage since 1982, I think also. So for here you have Islamic reasons, but also climatic reasons, because if you limit the opening to the hot and dry climate, so automatically you will save the energy, and you will save also the inner temperature from the outside environment. So here they use a lot of really small openings. We can just talk, call this windows. For me, it's more a small opening. Okay. So for the building form also, so you can see here, so sometimes we have the impression that the form is irregular, but in reality, they are irregular. So they just follow the shape of the side. And for the layout, for the plan of the house, I'm going to show you. So you can see here, you have heavy walls with local materials. So you have stones, you have sometimes brick walls with earthen material. And so I'm I'm going to show you later a little bit the plans of the houses. So for the orientation also, so they try to protect themselves from solar radiation. So most of the time you have the facade, which is almost blind, but for the patio, it's open to the sky. So they have luminosity, and they have also radiation, but in the inner part of the house. So the objective here, so you have shading, it's the priority here, but because if you have shading, you will have cooler spaces, and they try to also to increase humidity, the ventilation and cooling. So these are the objectives of courtyards, and it's not limited only to Saharan climate. So you can find also these specificities in old cities in North Africa. So I can show you here, you can see on the right side, this is the layout of the first floor and the second floor. So you can see here, you have in yellow, this is the courtyard, and all the rooms are looking on the courtyard. And in the first floor, you have like a gallery. So Mozambique call it ikomar. So this gallery ensure shading. So this way, all the rooms are shaded and they are cooler. And it's also for circulation also. So as you can see, just a second, you can see here the form. So it's not really regular. But if you see the next, the neighbors, you will have almost the same surface. So even if it's not regular, they really tried to be fair with all the community and to have almost always the same surface. Here you have a section. So as you can see here, we don't have domed roof or curved roof. Here you have flat roofs. So usually when you are in a hot climate, people try to avoid flat roof because for solar radiation. So because in this sense, the flat will be exposed directly to the sun. So they try to reduce sunniness and to do curved roof, and this way preserve the external surface from solar radiations. But here, no, they try to stick to the flat roof. And this was for the community. This way they can communicate between each other. So the roof here is used for women, and the women can just meet between the roofs, talk to each other. So the streets was for men, and the roofs were for women. So this way they preserve social relationships, and so men can meet in the streets and see each other and talk to each other, and for women they can meet on the roof and try to have the news of what is going on in the neighborhood. So it was a way so to keep the relationships, to keep also the family, to keep the neighbors. So it was really a community sense. So it wasn't only architectural or urban aspects. It was also social conception. So you can see here. So, and just when you can see in the middle, you have in the center of the house, you have really the heart. So the patio is the place where people meet, when they can have lunch together, dinner together, when they can rest, and also when they can also receive friends and neighbors and families in this space. So the central space of the house is really the core of this architectural conception. Just to give you an idea here, we try to see the temperature between external temperature and inner temperature to see if there is a difference. And if really this system is working, when we try to talk about thermal comfort, to see if really people inside these houses are feeling good and comfortable. So we try to do a small research to compare the temperature between inner and exterior spaces. And for instance, if we see the temperature in the streets and the courtyard, you will have the same temperature, because in this sense, you have, it's open to the sky. So the street is open to the sky, and also the patio is open to the sky. So usually you will have the same temperature. But when it comes to the rooms, when you come to the rooms, so here it will be different. I'm sorry, I think sorry, just for the rooms here, I thought that I had put a slide on this, apparently I deleted it. So for the temperature inside the rooms, if you compare the temperature inside the room and the patio, you will find the difference. And the difference is due to the gallery. So the gallery ensures shading, and with shading, you have cooler space, and the room is not directly exposed to the solar radiation. The gallery is working as a space which is between the patio and the room, and this way it ensures a fresh temperature to the room. So this was, and also the small openings. They are also located in a really smart position. So this way they can ensure a ventilation between the patio and the streets. So you have air circulation between the streets and the patio, but in a really discreet way. So for a passerby, you would not notice that there is here an opening that give directly on the patio. You will not notice this from the outside, but in the inner side here you will see that there is a difference between inner temperature and outer temperature due to the ventilation between exterior and interior. So it was really interesting to see this, because usually we just think that the patio is the main element, but we don't really focus on the small opening. So, but they are really helpful when it comes to decrease temperature. So I'm here for the thermal comfort. So for the heat dissipation, I took here a small example of a mosque because it was a curiosity for me. The mosque was built under the ground level. So I was curious, because usually the mosque, we can see it from the streets. It's not under the ground level. So I was curious to see how they built this mosque and why it is under the ground floor. And here I was, so here, I'm sorry, I will just show you here. So this is the structure. So this is the mosque. And as you can see, you have the minarets, and you have underground, you have the prayer place. So for the exterior part, it's just painted in white. And as you know, white color is good when we want to have some reflection, and also to give some freshness. But for the, I'm sorry. So this is the blind facade, open courtyard, just trees. I'm sorry, I went directly to the mosque and I far away. Okay, so for the trees, so just here some elements, additional elements, so as you can see from the external part. So here, for just a reminder, you have blind facades here, so you can see here in the minarets that is curved. You can see here also for the walls with the local materials and stones. And here also sometimes they use trees and vegetation, and also to decrease also the temperature. And now also I'm going to show you the mosque. So here you can see it's like a sink here. So look, you can see here for the ground, they use sand. So even if you go to the mosque 2 p.m. and it's very hot outside, it's really fresh inside, because the sand will is like a sink. And here it's become a natural element that decreases temperature inside the building. So instead of using, like now, they use cement and they use reinforced concrete, here they use natural materials. And the sand is having, has here a central element as a sink and to reduce the temperature. So you can see here, all this is underground, and you have just small openings on the side. So you can see also here another, it's the same mosque. You can see here, they try also to moderate the temperature here. So for the structure, they use an arch. So sometimes it depends. So in this case, for instance, it was a mosque. So they use an arch to reinforce the structure. But to limit the convection, you have the sand here okay. And for the, yes, and you have here, you can see just the stairs, also really small, and just to go outside the mosque. So here it's to increase the thermal comfort inside the mosque, and even for people who want to go to the mosque in summer period, for instance, where it's really hot, it's possible to stay in the mosque and to do the prayer, even if it's 50 Celsius degree outside. So it was a really nice experience to see this. So for the recent research, because I didn't went there for a while, so I tried to see if people still enjoy living in the city, and if they still feel comfortable if they in their houses, and also to see the conservation of the city. If people still use these materials, or now we have cement, we have wood, we have reinforced concretes. So I tried to see if people try to conserve the city as it was built, or if they try to be more modern in their way of to save the city. Yeah, I finished. Yeah. It was the last part of I tried to be fast. Okay, so just as a reminder here, you can see here the layout of the city, and you can see here the houses. So they have almost always the same surface everywhere, and you have you have here the souk in the lower part of the city and the mosque in the upper part of the city. And just we took some examples to see to see the plan of the house, to see if it's the same map or if they change it, the inner conception. And we found out that people didn't modify their houses. So if we take the first map or the first plan and we see the next one, we will have the same. So people didn't modify the inside. I didn't modify also the outside. This means that even if we are a modern epoch, people are still using these houses with the previous conception. So you can see here also the first one was the ground floor, and here you have the first floor. The house is still the same. They didn't modify anything. Also for the terrace, they didn't change anything. So then we have also a section here, nothing changed. So we tried for the facade, also you can see only the doors. So it's blind. So we tried to do a questionnaire just to see people if they modified the materials of construction, because the plan is still the same, facade is still the same, the section is still the same, but we wanted to see if the customs are still the same and if also the materials of construction are still the same. So it was the question to see really if they try to safeguard the city as it was, or if they try to modify it. And so the question was in French language. And just to give you an idea, I'm going directly to the main concern here. So most of the people live here since generations. So it's It's transmitted from generation to generation. So it wasn't the issue here. We have always the same community. So what we try to see for the material of conception. So for instance, here we try to see if people have a basement, because the site is not flat. So usually we'll have a basement. So we try to see, and 100% have a basement. We tried also to see if they have a well for water, and no one has a well in the house. So in this way, they still use the same way of living the space. Also, we try to see for the patio, if there is a modification, and it depends. Some 30%, for instance, 66% did modifications. So we tried to see what kind of modifications. And it was only modifications with the way of decorating the patio. So it wasn't really an issue against the form of the building, or the form of the patio, or the materials of construction. It was just a way of using the space. So it wasn't really a big deal in this sense. And you have here for the roof also, so 100%, they have flat roof. And we try to see if they still communicate between each other from the roof. And we have 66% who answered no, but 33 are still using the roof as a space that they can share with their neighbors. Also for the materials of construction, just a second. So here, so they try to see if the materials are still available. And we have a lot of people, they answered yes. So if they want to restore stone, it's available. If they want to use, for instance, palm tree, also it's available. But this is just, I would like to mention this, because when it comes to palm tree, it depends if the tree is alive or dead. So if it's alive, it's considered as a way to have food, to have shade, to have, so it's preserved as it is. But if it's dead, in this way, it is used for construction and it is used for houses. So palm tree is really important in Sahara. Okay, so this is the last, I think, slide. Also for the techniques, so this is the last one. For the techniques, we asked if they use cement, if they use woods, if they use concrete for their restoration. And we found out that they now don't respect really the natural restoration, and they try to include cement, and they try to include wood. Wood is not really natural over there. They were used to use palm tree, not other kind of wood. And also for cement, it's a new material of construction. They didn't use it before. So before they tried to have their own, like a recipe to do their own restoration with local materials. Now they're trying to adapt materials that are not original from the site. And they try now to include this in the building. And I think that they will destroy the thermal comfort, because cement is not for a Saharan climate. So it's better to use earth, to use sand, to be with natural materials instead of these modern materials that are not adapted to the Saharan climate. So
this was a really good final sentence. You said.
Conclusion.
We have really expanded our time. We want to have questions also, I think.
Yeah, how long I was?
You are longer than we had.
How much? Because I think so it's 15 minutes. Yes. So for the conclusion, I'm not going to read all this. Just a conclusion, because of the city of Ghardaya is, as I said, is a UNESCO heritage. So this research was more on the inhabitants and the way they live in the city. because they live here until the 11th century. So you have many generations living here. So we tried just to see if they still like the city, still enjoy the city, if the thermal comfort is guaranteed. So this was our priority. So, and from the architectural urban aspects, we saw that this city is sustainable because we don't use a lot of, how to say, climatiseur. Air conditioner. So it's really important. So when we want to save energy, this is a really good option to have a city a sustainable way and to use wind, to use sun, to use natural elements, and to include this in architecture. So thank you.
Wonderful. Thank you for taking us on the tour to the city, and please throw us open for questions. Okay.
I would say, first of all, it's not a question, I'm not a specialist, I'm super curious, just remarks. It's about the mirror of the people.
Yeah.
It's I think I'm wondering there is similarity with the in French in Iran. The wind tower.
No, it's not the same. No, they have wind tower, so they try to extract wind and to have fresh space. So it's not the same. It's no, it's not the same. Here it's just use it as a watchtower. So you can go on the upper side and see the whole city and the whole side, but they don't catch wind here. For the Middle East, it's a wind catcher. So they catch the wind and they use the wind inside and then extract the wind. So it's a wind catcher. So not here. No.
And I heard it's strange that they preferred social connection. Yes,
from the roof, yes, it's still used.
I believe that it's very, very hard. I was with hyphen in architecture in Jeddah. And the same thing, but they had not yes.
So they use the roof also.
Okay, the same conception in German. The same conception, the same stairs.
Okay.
Orientation.
Yeah.
On the street and everything, I think it's more than Phylogenetic of Tunis. Yeah. More than
for Tunis and for the capital city, for the Medina of Tunis, it's like the Medina of Algiers. So we call it the Casbah of Algiers. It's different. For the roof, for instance, they use this for the sacrifice of the Eid. So usually it was a way to see who did the sacrifice and who didn't. So, and they will share the meal with the neighbors. And it's a way to have fun and to have a feast as a community and not only as a family. So I know that for Algiers, for instance, for the Casbah, it was another tradition. But here no, here the roof is dedicated to women. So because
yes,
to speak to the men and telling the problems with
the house and everything. It's
white color, and they give it underground to go up fresh air.
Okay, so we have to go to Jeddah. Okay. Yeah, Ibadit, they came to Jerba and they came also to Algeria. So and they settled down in yes. So it is sustainable. Yes.
Yes,
Okay, thank you very much.
You're welcome.
Amazing presentation with very, very nice script architecture of the Gherdaya city. How many people are living now in Gherdaya? This is my first question. And when I saw the questionnaire, there are 3 responses, 2 responses.
How many?
Two answers, 3 answers in the questionnaire. So it's a number of people answering the questionnaire?
No. The questionnaire was for. Yes, because it was an online questionnaire. With the pandemic, we couldn't go over there. So the students, because I did this questionnaire with 1 master student. So she couldn't go to Gherdaya to do the questionnaire. So we prepared the questionnaire online and we send it to Google with Google Form. And we try to connect with the office, because there is the Office of Conservation of Gherdaya, and also with the touristic guides, and also with students, because we teach at the National School of Architecture Urbanism, and we have also students from there. So we try to have many contacts over there, and we try to send the questionnaire and to have, after the completed questionnaire, and to see how many people responded. But sometimes when you have a specific question, people try to avoid it, to be neutral. So they don't say yes, they don't say no, they try to be neutral. But we try to have some specific question, and sometimes you have someone who just write a line or 2 to make us know if he is satisfied or not.
So my question is the number of participants in this questionnaire,
yes.
Is it representative of the population of their land?
No, not the whole population, because if you take the city, now there is a problem over there, because people have just in oasis, they were used to have summer residence. And now they are looking to live in the summer residence. So sometimes you find the same family living in Gherdaya city, and also in the residential space. So you can't really have an exact number of the local census from my side, but because they did the last census in 2008. So what I show you, 311, it was, now they're just speculation of the number. And I don't have the exact number, so I can't give you an exact number of the exact number of inhabitants at the moment. But for the questionnaire, we try to send it to almost 100 people, but we get only, I think, only 50 responses. 50, yes. But we try to focus on 3 parts of the city. So the economic area, also the residential area, and the religious area. So we try to have this. But the main focus here was about the local materials and the thermal comfort. So it wasn't about the social community or the architectural aspects. Yeah, thank you. You're welcome.
Thank you, Dougga, and I think I will hand it over to Olga, who has some final.
Okay.